Now is coming in the full screen.
Yes, it's coming.
Yeah, it is coming now. Okay.
So,
Yes.
Yes. Supratim, shall we start?
Yes, Shriram,
Yeah.
please start by introducing.
Okay. Yeah. Hello, everyone. Welcome you all for this special lecture on this afternoon. Today we have Professor Subhadeep Chatterjee, who will be giving this special lecture on behalf of the Institute and iGEM student. And just to introduce in briefly, Professor Subhadeep Chatterjee is a staff scientist at CDFT, and he has done his education from Guru Nanak Dev University, where he did his MSc in biotechnology, and he did PhD from CCMB, and he did postdoc from UC Berkeley. And he has been working on understanding the mechanisms of host pathogen interaction, specifically with reference to Xanthomonas as a model. And he has discovered reversible non-genetic heterogeneity in bacterial social communication systems, coordinating virulence and lifestyle transition during plant disease. This is just a summary of what all his achievements, and he's also trying to apply this knowledge in engineering this pathogen to to control the plant diseases, so that to develop resistant varieties. And he has been publishing in very good journals like high impact journals such as PNAS, Cell Reports, PLOS Genetics, EMBO, PLOS Pathogen, and JXP, and he has several MPMI papers as well. And with these kind of papers, you can may imagine that like he has been awarded Shanti Swarup Bhatnagar Award in 2020 for science and technology in biological sciences, and he's also a fellow of all the three national academies, and he's been confirmed with Lalji Singh Memorial Award and National Bioscience Award for Career Development, and he's also elected member of a Guha Research Conference, and he also served as associate editor of Phytopathology Journal, which is published by American Phytopathological Society of USA, and he was also awarded Innovative Young Biotechnologist Award and Anindya Kumar Ghosh Memorial Award. And you can see he has numerous awards, and he has been doing some outstanding work ever since he's become a independent group leader. And with this introduction, I would like to request Professor Chatterjee to start his talk.
okay. So thank you all, particular iGEM team of ICER, Kolkata, Supratim and Sriram, and all the students who are involved in the iGEM, and also those who are in all the departments. So thank you for giving me an opportunity to speak about the bacterial lifestyle. So what I will do in this talk, I have divided my talk into basically four parts. So in the first part, I will talk about how we understand language of organism, and what we learn from their language, whether it can be translated into their regulation and their behavior and the social life. So then I will focus on the bacterial system, particularly pathogens, which communicate with their own language, and how they communicate, why they communicate. And then I will come to the model plant pathogen, Xanthomonas oryza, and what we learn from its understanding the biology of this pathogen. Because in my opinion, that if you understand the biology of the pathogen or its interaction with the host, then you can devise or you can come up with a strategy to prevent disease or interference of the disease. And at the end, I will talk about some of our applied work. These are all based on what we learned from a basic biology of the pathogen, and how we can apply. Is an example like how we can apply this understanding the basic biological knowledge in designing either disease resistance or interference of the disease. So if you see the communication, so the response by communication is one of the very key feature for all living organisms. Like all different criteria for a living organism like reproduction, growth, heredity, respond to the environment is one of the very crucial factor. Actually, if the organism don't respond to the environment and they can't communicate, so then there is a very disaster effect on the lifestyle of the organism. And if you see the communication at the highest level, if you see the languages, for example, communication by living most of the human race. For example, if you take just an example of English literature, so now you see this literature is very analogous to anthropology. If you see the anthropology people and also the evolutionary biologists, just like evolution of any organism, language also evolves, and it is all in a temporal and spatial pattern it evolves. So a very good example is the English language. Like if you go to different geographical region, the although the dialect is same, but many basic things changes, and that is to suit the local niche. So very similar, like languages are also very evolving, very similar to natural selection. If you see, go from language from the highly evolved organism to the very lower organisms, insects, animals, or any other small organism, so they also communicate with some language. For example, in the night, if you have seen that if the firefly blinks, so it blinks in a particular frequency and in a pattern. So actually, it is a call for its mating. So this is the language they use. The best example I can think of is the communication among the small invertebrate is in the honeybees. So honeybees they do a dancing movement. So this is called a dance language. Actually, they with that language they actually tell their fellow mates where the good nectar harboring flowers are there. So there are different auditory, olfactory, as well as different verbal and also the sound based communications. So these are all evolved, has been evolved for the fitness of the organism to survive under challenging environmental conditions. But if you come now to very tiny organism like the bacteria per se, like if you see the bacterial cell as such is hardly 2 to 3 micron in micrometer in length, and you can have to use a very high power device like electron microscope etc even to see it properly, and they are very tiny in nature. And a very burning question which has been arising till from the mid 1990s is that do bacteria also have a communication method, and with that communication method do they also have a social structure? Otherwise bacteria has been thought as a very lonely solitary living organism. So curiosity was driven by to the microbiologists as well as biological scientists that whether such a tiny organisms they also have certain kind of a language or a social structure. So all the clue actually came very indirectly from a squid which is called a Hawaiian squid. Okay, so this Hawaiian squid is found in Pacific, and this squid actually preys upon small shrimps, which is particularly are more vulnerable in the night. So what marine biologists who were working on this squid found that this squid actually uses a kind of light glows from its light organs, which are indicated in arrow, and that light actually helps the squid to find its prey. So they were got very interested and they wanted to find how this squid is producing this light and what is its role in the squid. Surprisingly, when they analyzed the squid in detail, they found that the light which is produced by the squid is not its own light. It is the light which is produced by millions of bacteria that colonizes the light organ of the squid. So they marine biology thought it's a nice symbiotic kind of relationship in which the squid harbors and gives a shelter to the bacteria, and bacteria in turn produces light that helps the squid to find the food. Then the microbiologists got interested why how this bacteria produces light. So they did an experiment. They started culturing the bacteria, and what they found that when the density of the bacteria is low, they don't produce light. They only produce light at a very high cell density. So that kind of interested them, and they did a very clever experiment. So what they did, they took the supernatant of the growing large density, high density bacteria, without the bacteria, just the supernatant, and they put it in a low density bacteria. And surprisingly, they found that the low density bacteria, which is not producing light, they started producing light. And that they gave them the clue that the high talking bacteria is producing some chemical communication or substance or message which is secreted by this bacteria. Then a whole field of socio-microbiology began, that people started to understand how bacteria exhibits and also does this social behavior. For example, they often forms biofilm, which are multicellular like structure, which you see on the right side. So actually these are mountain like shapes, but these are actually confocal images where millions of bacteria they form a super colony or a housing society like structure. So and that helps them the bacteria to withstand defense response of the host or to the changing environment condition. So this transition from a solitary to the biofilm and then the vice versa is kind of a regulated by a process which is called as quorum sensing. So in prokaryotes like bacteria, they secrete small signaling molecules which are depicted in red. So when the density of the bacteria is low, the amount of the signaling molecule is also low. And when the density becomes high, the amount concentration of the signaling molecule also increases. And when it reaches a particular threshold, it tells the bacteria what now you should do. It basically synchronizes bacteria to perform certain tasks in unison. And those tasks may include production of enzymes to destroy the host or to attack the host or to carry out biofilm formation. So in the lab, basically you cannot listen to their language, but you can see the language. So what we use is different type of biosensors, which you by the use of this biosensor, basically you can detect different type of bacterial signaling molecules like communication molecules. For example, plate on the left and right side are filled with bacteria, but on the left side there is a reporter which is coming up because the bacteria are talking, but in the right side you see lots of bacteria, still they are not able to speak. So this kind of behavior, the unison behavior, is called as quorum sensing. Now, what are these language molecules? So it's a very complex language system which the bacteria utilizes. Just like the human race, like they have different languages. Some are public language and some are private language. So they also have different types of languages. The most common, well-studied language system is the acyl homoserine lactone mediated quorum sensing language. So where it is very, if you see the analogy of this language with our modern language, it is just like Indian language. Like Sanskrit is the base, the lactone is the base, and different dialects are added in the form of side chain, like different languages, Indian languages, to make variations. So I will come in the second part of my talk about a family of signaling molecule which a xanthomonas group of plant pathogens make, which is called a DSF, diffusible signal factor. So these are also a family of signaling molecules. Now, what is the nature of these signaling molecules and whether bacteria can make only one type of language or they can speak in multiple language? Actually, people got very excited that some bacteria, particularly pathogens, they can speak in different language. For example, at the right side you can see the two bacteria sitting. They are actually cholera, vibrio cholera, which causes cholera, a very bad disease, and they talk in two different languages. One on the top by a specific acyl homoserine lactone, that is to talk with each other, and another is a borofuranol compound, and that is for talking with their neighbors. So actually bacteria is so clever that they also utilize their private language to build up their own strategy, and they use a common language to know what the others are thinking and in the neighborhood. And some bacteria are very clever, so they don't make any language, but they have receptors through which they can listen to other bacteria which are there in the gut or in the host or wherever in the environment. Very similar to a noisy neighborhood cartoon in which if a new neighbor comes in, the other neighbors are more curious who has come, because in that way they can change their strategy and also the actual environmental interaction, how they can strategize there to best utilize the resources. So one burning question which comes is that why so much of effort that bacteria takes to make such a costly signaling molecule, and what is actually its benefit? So if you see this is a very simplistic cartoon. It's not is very complex, but I have represented it in a very simplified form. Basically, the bacteria, different bacteria have synthase, which is called a Luxi. So this synthase actually catalyzes the formation of the signaling molecule in a auto feedback loop, and as the density increases, more and more amount of signaling molecules are made because number of bacteria is more, and it reaches a particular threshold at high cell density at the right side, if you see, and then it binds to the regulators which are there, and it is a very concentration dependent process. So when the concentration has reached a particular threshold, then only the response regulator will be bounded, and that will orchestrate the gene expression in the bacteria to perform tasks which are done in high cell density, and certain functions which bacteria do at low cell density based on the low amount of signal. In that way, it actually bacteria can count each other and to determine what next task is for them. Now, a very important question in quorum sensing is why the bacteria do quorum sensing and what is the advantage in that. So I will give you an example in a very layman's term. Like imagine there is a person who is firing a bullet to break the wall. Imagine that is a cell membrane or cell wall. So the firing power of the one bacteria or the one person will be so less that the wall may not break, and the nutrients which are released after breaking the wall will be not available. So it is a wastage of the resources to fire alone. But when lots of bacteria are there and they together they fire, the firing power is so concentrated that there is a great chance they will break the wall and the benefit will be more. So this is the whole concept of quorum sensing, that in quorum means if you do certain task in unison, the benefits of the return or the benefits, the fitness effect will be much more. No matter you when you are alone, you can do that same task, but the benefits will be less. So this is the whole concept of quorum sensing. So in our lab, we work on this model pathogen Xanthomonas. It's a yellow colored bacteria, produces copious amount of extracellular polysaccharide, and this yellow color is due to the production of a pigment called as xanthomonadin, and this pigment actually protects the bacteria from UV lights, so it acts as a sunscreen. And Xanthomonas is a very important plant pathogen because it has a very wide host range. Around 350 different plant diseases are caused by Xanthomonas. This includes rice, many vegetables, fruits. So in our lab, we mainly concentrate on the rice pathogens, called as Xanthomonas oryza, which is a vascular pathogen of rice, and another is Xanthomonas oryzicola, which is a non-vascular. It's a tissue pathogen. So both has a different mode of entry and tissue specificity. We also work on Xanthomonas campestris, which is also a Xanthomonas which particularly attacks lot of crucifers, for example, cabbage or mustard and other crops. So, what to cut a long story short, I am not giving you because the time is limited. So working on the basic biology of this Xanthomonas, what we encountered in couple of years, some years back, is that this bacteria actually produces a signal which we named as diffusible signal factor. So it is a fatty acid like signaling molecule. It is a cis-enoic fatty acid, which is quite unusual in the bacteria. Bacteria generally produce trans fatty acid and it's a secreted fatty acid. And what we found that if you have a knockout of this fatty acid, this synthase, you completely lose virulence. On the right side, these are the rice leaves which are infected with Xanthomonas. At the extreme left is infected with the wild type, and you can see the browning of the leaves. And after 10 to 14 days, the whole plant dies. And you see the mutant in the middle when it has been inoculated that cannot produce DSF signaling molecule. It is not at all able to cause disease. It stays wherever it is. And when you either put back the wild type or you supplement it with the DSF exogenously, the mutant regains its virulence. So we wondered like how a quorum sensing signaling system will be important, how it is affecting the virulence. Then we started to look in the lifestyle of the Xanthomonas, and we found that is actually it colonizes the xylem vessel of the plants. So in the red pipe-like structure which you are seeing are the vessels which can conduct water and ions, and this bacteria actually forms a clusters or biofilm which are depicted in green, and they are colonizing. It's a biofilm like community. And what we found out, these quorum sensing deficient mutants, they spread throughout and they don't form a biofilm inside the xylem vessel. Then through many genetics and screens, we found out that it has a very complicated lifestyle. If you see the lifestyle of a plant pathogen, in general, the foliar pathogen, on the top it's a cartoon of a leaf structure, and if you see this bacteria is moving around on the leaf surface, and there are some openings which are like fountains which it has to enter to get food. Imagine the size of the bacteria and the size of the leaf. There is a huge difference. It's in micron, it is in centimeter. Okay. So now, if you see, if the bacteria does a random movement to find the door by chance, it will require its lifetime of its the whole period to find the spot. So therefore, the model is like at low cell density when the numbers are less, because at the initial stage of infection, when the bacterial numbers are less, so the low cell density may trigger a chemotaxis mediated movement that will help them to find the niche or the entry points. And once they enter, they should have an active motility component which might be activated and to spread throughout the xylem vessel. And at that point, they also have to secrete cell wall hydrolytic enzyme to get nutrients, to break plant cells and to get the nutrients. And when more nutrients are released, their cell number more increases because of more growth. At that time, the plant defense response they had to counter, and they at that point they trigger the formation of biofilm by a very orchestral synthesis of adhesins. Adhesins are attachment proteins which are made in a very spatial and temporal fashion, so that right time the bacteria has to adhere and to each other as well as to the host surface to form a biofilm in order to get more nutrient and protection from the plant defense response. So this is just an example of an adhesin. You see this mutant which is called RPFF, which is a DSF deficient mutant. It is always planktonic, and if you see the wild type, it forms a nice biofilm, and you can overcome this biofilm problem by adding either DSF exogenously or by expressing the DSF synthase. So after we started interested in the quorum sensing biology, we figured out actually many adhesins which are actually made in a very concentration dependent fashion in the xanthomonas. So there are certain adhesins which are required at the entry point to anchor at the entry point. And then as the density increases, they have to be different type of adhesins are required. For that, it will facilitate its spread. And when a critical concentration is reached, they actually makes three different type of adhesins which we identified, and we name it as a ZM, ZY, Z meaning xanthomonas adhesin M. So they are made in a very orchestral fashion, so that to synchronize the infection stage. So at right time they are made so that they can anchor, and also they help to promote their spread. So then with to understand the biology of the signal transduction process, I am not going much into detail, but this is just to give you a glimpse that as like human, their signal perception and their language perception is very complex. It involves several transcription factor response regulators which on the course of our work In CDFD, we have identified each of them through many either forward or reverse genetic screen. So if some people are interested, we can talk at the end of the talk because of the time limitation. So in general, the take-home message from this thing is that in general, as I told you, there is a synthase which is called RPFF, which makes the DSF, and it forms a complex with the response sensor, which is a hybrid two component sensor called a RPFC, and then with the crosstalk with different transcription factor and environmental sensors, it undergoes the regulation in a two stage. One in a DNA binding mediated regulation, and another is an allosteric regulation. So the allosteric regulation happens by synthesis of a intracellular signal which is called as cyclic di-GMP. So that comes in the second hierarchy. And in the first hierarchy, actually, this transcription factor called RPFG, CLIP, and sigma 54, they actually may further subregulons to further regulate the expression of genes in response to the quorum sensing. So by making it in a simplistic model, so what happens at the low cell density at the entry point at the right side? So the bacterial concentration are less. At the low concentration of quorum, actually what we have seen that it activates the chemotaxis and motility system in this bacteria, and that helps them to guide and enter into the xylem vessel through openings which are called hydrathodes. And once they enter, they slowly build up their population by the whatever the residual nutrients which are there in the xylem vessel, and they start secreting the green bullets which are called as cell wall hydrolytic enzyme. And this hydrolysis of local plant cells release a lot of nutrients, and that help the bacteria to grow. And then once it grows a particular threshold, it the quorum sensing signal also builds, and it tells the bacteria now you have to form your own home. And in this way, it makes an orchestral of this adhesins in a spatial and temporal fashion to make a home for it. But the biggest question in this bacterial biology, and also in the plant, also in the host pathogen biology, then what happens next? Imagine a situation if there is a biofilm formation and then it started growing, but the bacteria also has to detach from that to find the new niche. So how this reversibility happens? So what we found out in our lab that this bacteria actually exhibits a reversible non-genetic heterogeneity in quorum sensing. So reversible because it is either high quorum or low quorum, the cell can decide, and there is a stochastic distribution. And it is non-genetic because to maintain the ratio of the performers and non-performer, that means the quorum plus and quorum minus cells. So I will talk about why this is very important in the biology. So as I was telling you about mechanism of quorum sensing, how they perform signal transduction, and then how they utilize the signal synchronization to perform social tasks in unison. But every process has both pros and cons. So actually, if you see that talking is a double-edged sword. So what happens? Because see the panel at the left-hand side top, you see a population of bacteria which is now talking and performing social tasks. Imagine a situation in that certain cheaters arise. They may be a genetic mutants or maybe a random mutants which have arise, which are not able to communicate. But they are very happy with the population because the other people are performing. Very similar exam. So if you see the examination is going on, the many people have prepared, but there are certain cheaters who has not worked, which are taking the benefit from the performer. So if the system keeps on going like this, what will happen? Soon the classroom will be filled with cheaters, because performers will also lose lot of this energy in doing the social task, and then there will be more preference for selection of cheaters. So this is one problem of quorum sensing. And the second most important problem of quorum sensing is that too much of talking is also very bad, because in that way you can alert the host defense response. So now in the third part of my talk, I will talk you how this bacteria cleverly deals with these two double-edged sword and still maintains fitness. So if you see the quorum sensing which I was talking, that it synchronizes the cell to perform social tasks. And I told you the problem can arise that if you everybody is performing, then there is a chance of selection of a cheater. But what evolutionary biologists and also the experimental evolution people, they suggest that phenotypic heterogeneity in performing social tasks is also sometimes advantageous, because it serves as a bet hedging survival strategy. Okay, I will give you an example in a layman's language. For example, in a family of four, there is one bread earner and the other person helps in the household activities. Third person also helps sometimes, uh, help the first bread earner or the domestic work, but the fourth member doesn't do anything. Okay, so he is very computer savvy, and he works always games and plays with his computer. Now what is happening in the house, it is supported by the action or performance of the three individuals. The fourth is enjoying, and the system is going on. But suddenly what happens, for example, COVID like situation comes where there is a work from home. Now the bread earner has to work from home, and he has to work with his computer. Now suddenly one day his computer fails. Now uh, uh, the fourth person who has not contributed anything for the family per se, now he quickly comes and fixes the computer of the bread earner, because there is a benefit, because he also has a benefit, because if the bread earner doesn't earn, then he will also not get food. So he contributes at a particular condition. So this is what is called a bet hedging. So actually in the population there are phenotypic heterogeneity is maintained to, so that sudden change in environmental condition or sudden change in situation may cause sudden wipeout of all the population. But if you have variants in already existing in the population and which is reversible, then there is a great chance that the population will survive. And basically, this is how the bacteria avoids this tragedy of commons. That means the balance between cooperation, bet hedging, and cheating. And so, this is what we have given a model that bacteria maintains reversible non-genetic heterogeneity, maintains two different type of population, both in vitro as well as in vivo. We have shown that when bacteria infects, so there is a 80% of the population which are quorum sensing, but it keeps switching on and off 20% of the population, which are depicted in blue, which actually doesn't sense the quorum transiently in 4 is to 1 ratio, so that they can escape the biofilm and again give rise to 80-20 proportion of the population, so that the system goes on and there is a maintenance of heterogeneity, maintains stability. and there is an environmental switch, which is the nutrient, which actually controls this switch that it never crosses 80-20. Because if there is an imbalance of 80-20, then there is a fitness cost to the cheater. So I talked about how the bacteria encounters the first problem of cheating and cooperation. The second problem, like alerting the host, as I was telling, that too much of talking maybe also cause alarm in the host. What we found out that this Xanthomonas, when it interacts with the plant, definitely they perform quorum and they talk with each other loudly. But what happens in the course of evolution that plants have developed the receptors or the signaling mechanism to detect bacterial signaling molecule, the quorum sensing signaling molecule, as PAMPs. Actually, our lab first time showed that a bacterial quorum sensing signaling molecule can act as a PAMP in a plant, and how and it mounts a defense response. Then one may wonder then how then infection happens, because bacteria is one step clever. It secretes an effector that again suppresses the host innate immunity system. So that we have shown in a cartoon form. Because of the lack of time, I'm not able to show all the data. But what happens during the entry point, what happens, the bacteria are low in number, so there is a less amount of quorum sensing signal, and low amount of quorum sensing signal actually primes the plant to prepare for a larger defense response. And once the concentration increases more in the concentration increases of the quorum sensing signal molecule also increases, and it triggers the early defense response, that is stage 2. But at that point the bacteria start secretes a effector which is also controlled by quorum sensing to silence the plant defense response. And this effector which we identified is extracellular polysaccharide. Actually it chelates calcium, which is an important trigger for the plant defense response. So it lowers down the calcium spike. So this is in a very nutshell what happens. The bacteria and the oryza and the plant interaction. So the bacteria interacts with the plant with the type 3 secretion. It has various type of secretion system, and it start producing the quorum sensing signaling molecule, which is depicted in red balls. And as the concentration increases, and it helps the bacteria to perform social tasks, which I showed you earlier. And by the course of production of this signaling molecule, these signaling molecules are now detected by the plants, which are called a PAMP receptor, and it induces a MAP kinase signaling pathway. I have not gone much into detail. We have identified many components of the plant component, host component, which are involved in the perception of the DSL, and it induces a innate immunity response by ROS, reactive oxygen species, callose deposition, and calcium burst. And this pathogen also makes polysaccharide, as I told you, extracellular polysaccharide that chelates calcium and dampens the plant innate immunity. So it's a two-way war which is constantly going on in the nature. So the war between the pathogen and the host, and of course the pathogen has the advantage because the doubling time, the multiplication time of the pathogen is much higher than the plant, and you always get variants. So now I will come to a different strategy, like what we learned from our basic biology of a pathogen and a host, whether we can devise some strategy to interfere with the disease. Sometime back, a very interesting article came in Scientific American. So it is telling about a weakness in the bacterial fortes. So what it's telling that instead of targeting a single target, like for example, we use several antibiotics, right? And the antibiotics are very specific, so they act on a specific target. Now, suppose if you have a mutation in a specific target, trimethoprim, or let's say nalidixic acid, which target gyrase, DNA gyrase. Now you have a mutation, you get null resistant colonies, and this is the major problem in hospitals as well as in, if you want to do any biocontrol, because the resistance is so quick in the microorganism that you will get a resistance. So what they are suggesting is that instead of designing inhibitory molecules or target antibiotics for specific targets, so you design molecules or targets to their social structure. That means if you can poison their social communication, then it will break everything. Because if you have a mutation, counter mutation in the pathogen, then it is a cost for itself. Then soon it will be outcompeted by the wild type. And if you have the molecule that can block the communication, then the whole several downstream pathway can be blocked. So one of the example is has been tried in US also, is about the iron nutrition. So what happens? Many pathogenic bacteria, they require iron from the host, and they secrete. This is an example they have tried in hospitals with Pseudomonas aeruginosa. So Pseudomonas aeruginosa is a opportunistic pathogen, particularly in cystic fibrosis patient and those patients which are immunocompromised, and it colonizes the lungs, and soon the whole lungs is covered by the bacteria, and it's very difficult to remove because they are multi-drug tolerance. So what they have found out, this iron uptake and iron homeostasis is very important for this bacteria. So these bacteria secretes hydrophores, which are depicted in green color balls, and this hydrophore actually sequester iron and takes away iron from the host, and that hydrophore is utilized by the Pseudomonas. Now what they have found, that if they put gallium, which is an analog of iron, now gallium when bind with the hydrophore, but the gallium bound iron cannot be taken in by the bacteria. So it basically actually poisons the bacteria, and production of hydrophore is also a quorum sensing regulated. So if the enough amount of iron is not available, the bacteria is will be wiped out from the, and they got very good results in severe Pseudomonas aeruginosa infection. So in case we thought, in case of quorum sensing, we can also utilize this kind of strategy. So one way is by degrading the signaling molecule. If you have an enzyme, or you can engineer an enzyme which can degrade the signaling molecule, then there is no communication. The second approach is what I call it as a loudspeaker approach. So now, as I told you, the concentration is very important for this proper communication. For example, if you, this is an, imagine a situation, if there is few people talking with each other and you play a loudspeaker or music, so they are not, they will be then not able to communicate. Exactly like that, the second strategy we followed, we thought that if we make the quorum sensing signaling molecule in very high amount, either in the host or ectopically, then we can probably break the actual communication signaling, because no meaningful communication may not happen. So we used both a transgenic and non-transgenic approach. So this is to manage diseases by interfering with the cell signaling. They are nowadays called as social drugs, because you are targeting the social life of the bacteria, both in plants and animal health. So to cut a long story short, actually it was a lot of work. So we designed the whole bacterial operon, codon optimized it, and make a regulatory circuit so that it can make the bacterial signaling molecule, and codon optimized to be expressed in the plant. And in the plant, since bacteria are close relative of chloroplast and mitochondria, so we thought better to target it in the chloroplast. So we targeted this thing in the chloroplast and developed transgenic plants which are now able to make lot of bacterial quorum sensing signaling molecule from the beginning. And then we did an infection assay. So if you see, the y-axis is the severity of disease. So more the severe means more number of leaves average are infected. So at the right side, extreme left side, so this is the plant, the wild type plant. You see, it's a highly affected by the disease. All the transgenic plants, what we tried, which are expressing the bacterial quorum sensing signaling molecule, and we showed also that the transgenic plants by LCMS and also other quantification, that now they started producing the bacterial signaling molecule. So they are very reduced in disease. So what is happening is that when bacteria is infecting this transgenic plant, even though they are low in number, they are seeing that they are too high in number, because actually the message of high number is given by the plant, and they are thinking now, okay, now we are very high in number, we will form a biofilm. Basically they are forming a biofilm at the site of infection itself and not moving inside the plant and spreading. So that we showed that in the transgenic plants, actually the wild type stops near the site of infection because it sees too much of communication that tells them that the bacterial numbers are very high, and you stop and make a biofilm like at the point of inoculation. But however, in India, particularly the genetically modified crops are not that encouraged. So we thought of developing a other strategy. So we engineered bacteria and other high producing strains to overproduce this signaling molecule in the lab. So basically you design the operon in such a way that they produce a large amount and in a good host, and you produce huge amount of signal, and that we can mix it with a surfactant as you see in the right side, and surfactant actually helps the chemical or a compound to penetrate and also to stay on the, because the plant surfaces are waxy, so applications of any chemical or water is very difficult, so you use very low percentage of surfactant, and then the results can be seen on the left lower panel. So there's a, at the extreme left there's a leaf which has been infected with which has been sprayed with the DSF, the synthetically the laboratory produced DSF, and then in the middle it has been sprayed with buffer, and then it has been inoculated challenged with the pathogen. Now you see in the buffer sprayed a plant, you see very nice infection coming up even after 10 to 14 days. But you see the pre-primed or the pre-inoculated, pre-sprayed with the exogenous DSF, there is a drastic reduction of disease. So now we are in talks with Directorate of Rice Research and ICR to scale up and also to use more field trials, so that if we can give up some formulation, so that it's a totally non-transgenic approach, so by the synthetic DSF and formulation it with the proper delivery. So this is at the last We also do a lot of this communication study using single cell and also the tracking of the communication. Now, what we see is that the bacteria communicates, which is an indication of the green fluorescence, and now you see the heterogeneity in the features which are coming up, and but the system always remains. So we may devise, but after some time, the bacteria are more than us. And thanks for listening the talk. So this is our CDF in Hyderabad. The names which are indicated in yellow, they are the current lab members. We're working on different projects. Actually, we work on xanthomonas, lysine, then virulence factor, quorum sensing is one of the part, and host responses. And also we are interested in chemotaxis and iron uptake mechanisms, and in the in at the new site there are X members whose life they have contributed significantly, and we are grateful to EBT, SARB, PSR for funding, and we also get support from CTUD. Thank you.
Okay, I think, am I audible?
Yeah.
Okay. Yeah, I think with that, let's thank Subhadeep for a wonderful talk. I hope all the students they enjoyed. If you have any questions, you can ask Subhadeep. Students, Tapas.
I have questions,
Yeah.
but hi, Subhadeep.
Hi.
Yeah. But I think students should get the priority. Let's see.
Yeah, students can ask.
Hello, sir. Sir, I have just
Yes,
a
Avijit.
little doubt. Like, uh,
I think there
in
is
the
a
case of
echo
Quran,
sound.
Hi, Sriram.
Okay.
You mute, then echo will go.
so you said about the lux, bacteria produce luxi, which first diffuse out, and when the density of the
Protein
bacteria.
doesn't diffuse, the signal diffuses.
Oh, yes, sir.
Yeah.
So luxi actually diffuse out from the cell.
Peroxide doesn't diffuse it. It's a protein. It's a biosynthetic protein. It makes the acyl homoserine lactone that is diffusible because it's a fatty acid and a acyl chain.
Okay, so actually it facilitates a reaction that produce acyl homocytyl lactone
Yes,
that
correct.
diffuse out and then come back again to
Hmm.
activate.
Because it's diffusible. Yeah.
Okay, so this was my doubt, and there is another doubt. Like in the last experiment, you said that treating with the DSF, the infection levels goes down. There also one more data like treating with cellulose. Also, I see that the infection level.
Yeah, because that is also a PAMP.
Okay, so
Cellulose
cellulose also.
is actually a damage associated molecular pattern. So cellulase actually people have seen that it acts as a damp, and the problem with cellulase is that it's a protein. An application of protein in the plants is quite difficult. So this is a small compound, is applied easily.
Yes, sir. Thank you, sir.
Say any questions?
Yeah, I have, but if students
Yeah.
have no other questions, I also work on biofilm, but not at that extent, not that extensive what you do. But before I ask my question, I did a fantastic work, and explanation also very, very nice. So I really appreciate. And the thing is that for Vibrio cholerae, what was found, that is high quorum when they produce, they flush out, it helps. But in your case, you are telling your
Yeah, actually, the
yeah.
Vibrio is a different biology. So it is quorum act as an anti-virulence type of factor. So if you have more quorum, Actually, that is actually helps in the, that is because of the lifestyle of the pathogen. Because in for example, in the gut or this thing, so it helps. It is much better not to form a biofilm, because if you form a biofilm, you are restricted. Then it's targeted,
Yeah.
the host defense response. So in most of the bacteria, quite quorum sensing can give about two different things. So in Vibrio system, what people have done in a different strategy, what they have done is a quorum kind of a mimicking compounds which can block the attachment and other things.
And what I found in case of Pseudomonas or other bacteria,
Hmm.
so if you go normally in a second culture, they are totally planktonic.
Hmm.
They can swim through.
Hmm.
But if you grow on a plate, they swarm
Yeah, yeah,
and
yeah,
they really switch in terms of their number of flagella.
yeah,
But
yeah,
if you provide them a kind of a liquid medium in front of them, like what these
yeah, yeah,
Xanthomonas do, they make it 3D biofilm
yeah,
structure.
yeah, yeah.
And we found this all actually manifested by means of differential reactive oxygen species
Ah, that can be
concentration.
possible. Yeah.
But is it, I mean, did you check in your system is the same thing happen?
Actually, the thing is in Xanthomonas, Oryza doesn't exhibit much swarming. So it does swimming and twitching. Twitching is by the type 4 pili.
Type of pili.
Yeah, type 4 pili. But in other bacteria, it is true that a lot of environmental condition also influences multiflagellation and this thing. So this Xanthomonas is uniflagellated, but in Pseudomonas or other, multiflagellation can happen. So the multiflagellation definitely is affected by oxidations, oxidizing and anaerobic regulation. And other thing, what happens in the planktonic culture is that when you shake, na, lot of
Yeah.
flagella get shredded out.
Yes. Yes.
That happens.
And another question, Xanthomonas, it produces some mucoid, some
Huh.
polysaccharide coating.
Huh.
So it helps the biofilm formation or it
Yeah, yeah.
saves.
Actually, I
Yeah.
showed you in the cartoon.
Maybe I
That
missed it.
actually,
I'm sorry. Yeah.
yeah, actually, it is one of the very important factor in the biofilm formation in the later stages. So what happens? The polysaccharide actually helps the attachment. So it's a three-way interaction. So bacteria makes adhesin. That adhesin helps to attach it with the polysaccharide, and then there is a polysaccharide helps to attach it with the host surface.
Yes.
And also it has a dual role. So it somehow protects the bacteria against the phenolics or the helps the bacteria to form biofilm, and also act as a suppressor of the plant innate
Immune system.
immune
So
system.
is it the same thing you are expressing in terms of exopolysaccharide, that part of the EPS, or it is different? If it is different, so does it also under the control of quorum sensing? There's
Yeah,
no.
it is under the control of quorum sensing.
Okay.
So, yeah, yeah. So now,
Okay.
so very cleverly, the bacteria has designed its weapon also based on its own need. So when the quorum is high, then it switches. The,
Switches, yeah. Okay, thank you. Yeah.
yeah, thanks.
Hello.
Hello. Yeah.
So can we tell this signaling as juxtacrine signaling?
Pardon? Can I?
Can we tell this kind of signaling as juxtacrine signaling, the quorum sensing signaling?
Which signaling? I'm not very clear. Can you? Which signaling you are
She
talking?
is telling some juxtacrine.
Just
Yes, yes.
Juxtacrine.
what is that? I'm also hearing for the first time.
I'm also hearing first time. Yeah.
What is
It's
that
like
kind of
paracrine
signaling?
and
Can you
juxtacrine.
explain a little bit?
Organism secretes some molecule, signaling molecule, and that binds to very nearer organism and
Okay,
run
okay.
some
That's a
signaling.
very good question actually. So whether it's a local effect or a broader effect. So actually, people have seen that quorum sensing is also diffusion sensing. So suppose there is a group of bacteria which is making quorum sensing signaling molecule. Obviously, the local concentration will be much higher, And since it's a diffusible molecule, it forms a gradient. I showed you in the cartoon, it forms a kind of a gradient. And those bacteria which are at the end of the gradient, they will receive less signal as compared to where the there are low. So there is a local effect and a distant temporal effect also. Is it have cleared your question?
Yes. Yes.
Subhadeep, I have a simple question. One.
Yeah, tell me.
I just want to know, like
Yeah.
this DSF diffusible signal factor, is it a single molecule or a group of molecules?
It's a single molecule, but I told you that depending upon the language, actually bacteria
Okay.
varies. Now this is a language molecule of Xanthomonas oryza.
Mhm.
Now it uses another dialect like Xanthomonas campestris modifies the acyl
Okay.
chain, but the core is same.
Okay.
The core is
Okay.
same.
Thank you.
Hello.
Hi, hello.
Yes, sir. So, sir, like,
Yeah,
how
go
do
ahead.
these Xanthomonas, yeah, how do these Xanthomonas distinguishes which language to or which chemical of foreign sensing to choose while they attack or while they communicate?
I'm not
Like, what
clear
is the
about it. Can you
is there
speak
any
it a little loudly?
so like what is there any how do they distinguish between the language that foreign sensing chemical while
Okay, so the
these
question is
Xanthomonas
how
attack?
bacteria can distinguish between two different languages or
Yes,
different
yes.
languages. So actually, the way it distinguishes different languages is that they have different receptors. Okay, so in Xanthomonas, there is a DSF receptor, which is I showed you that we found out that it's called as RPFC. But in other bacteria, they maybe have another receptor which can recognize their signaling molecule. But certain bacteria are very clever. They make different type of receptors so that they can communicate with other bacteria also. For example, in Xanthomonas, they have a DSF receptor as well as an orphan acyl homoserine lactone receptor. Although Xanthomonas doesn't make acyl homoserine lactone, but it has a receptor in it. And it's still $1 million question what it senses with that. Maybe it is sensing some other bacteria which is communicating with acyl homoserine lactone, which is also a plant colonist.
Okay. And under hello.
So is it your hello?
Yes, sir. So under a
Hel-
different, if you put some selection pressure,
Selection pressure on whom?
will they change their language or will they be able to change the
See, if you put, see, this
on
has
the
been
xanthoma?
selected. So, selection pressure, what kind of select? So, one type of selection
Like
pressure
any
is the
kind of stress?
cheaters. Yeah,
Okay,
stress if you do, but the language doesn't change. It only selects
okay,
for the fittest. So, among the population, it is very clear in the evolution. There is no screening. There is a selection. So what happens when
hmm,
you do very different type of stress, then among that population which is showing the heterogeneity and particular subpopulation which can better respond to that stress will be obviously be selected. But selection of a different language, so the whole receptor has to be changed and then
yes,
whole synthesis has to be changed. But one possibility is
okay,
that during the environmental interaction, what happens in bacteria, there are horizontal gene transfer happens.
transfer.
So one type of quorum sensing signaling molecule system can transfer in, for example, in Vibrio cholera. So now in Vibrio cholera, two type of quorum sensing system is active.
Okay, okay, sir.
So, Shubhadeep, I have a quick question.
Yeah, yeah.
So I think you've sort of answered this, but when you think of interrupting the quorum sensing as a method of therapeutic intervention, we also have bacteria in us who are helpful to us.
Yeah,
So there is this risk of
definitely.
okay. So one has to then engineer. I mean, look at what other kinds of bacteria one may.
Definitely, the whole study has to be done. What are the other types of bacteria? And particularly the advantage of quorum sensing is that different bacteria makes usually different type of molecule. But if you have a quorum alike bacteria which is also sitting there, then it may be a problem. But usually, for example, as I was telling you at the beginning, that although the core may be same, but the dialects are different. So other plant associated with that has to be seen, the effect. But it is maybe a because under laboratory condition you just prevent the disease. So what would be its consequence in a larger scale, like that has to be seen, ecological consequence. So these are definitely, but one thing is good that you can use this thing non-genetically also.
Right.
For example, people do nowadays lots of these microbial formulations to have a suppressive soil or maybe to prevent disease. But again, there may be cause or consequence of it may disturb the internal microflora or maybe
Microbial dynamics. Yes.
local, yeah.
Thank you. It was a very interesting talk.
Thanks.
Okay, I think we'll stop here.
Okay,
Yeah.
Sriram.
So with
Thanks.
that, I think, yeah, thank you, Shubhajit, for a wonderful
Thanks.
talk. Yeah, thank you for your time.
Yeah, sure. Thanks.
And they need a
Bye.
group photo.
Sir,
Yeah, I think somebody
Yes, sir.
Yeah,
wanted a
Chirag.
photo, right? Chirag is there? Or anybody
Or
else
anyone
can
from
take.
iGEM team.
iGEM,
Yes, sir, I can do. I'm ready.
Chirag, take photo.
Yeah, please show me the camera. I will take it. All of you can switch on your camera. I will take
Yeah,
it.
everyone, just put your camera
Yeah.
on. Chirag is going to take a group photo.
Done.
Thank you, Chair.
Okay. Bye.
Okay.
Thank you, Shubhadeep, again. Yeah, thank you.
Shubhajit, I have one question. Actually, the students wanted a record of your talk.
No
I
problem.
recorded. Is that okay? Okay.
Yeah,
Yeah.
no issue.
Okay, thank you.